Right now, if you have a cough, shortness of breath, or a sore throat, you're encouraged to get tested in case it's COVID-19. But what if it's something else? A U of R biology professor is hoping to develop a way to find out exactly what's going on. Andrew Cameron is also co-director of the Genome Prairie's COVID-19 Rapid Regional Response Project. And he joins me now. Thanks for being here.
Thank you for having me.
So why do you want to find out if someone doesn't have COVID-19, but maybe other viruses?
I will provide a two-part answer to that. First off, because COVID is not the only, or SARS-CoV-2 is not the only virus circulating at the moment, and certainly won't be the only virus circulating this fall and this winter. So we are always experiencing the potential for overlapping outbreaks of different viruses that can cause infections. And what could be really bad is if you get two of those viruses or more, perhaps at the same time. We are not just testing for COVID. We will be testing people who have COVID to see what additionally they may be infected with.
So what's your plan? How does it work?
Our plan is to take a very broad approach. So currently, if you go in for a COVID test, they take that swab from the back of your nose and they look for two very specific parts of the genetic material that make up the SARS-CoV-2 virus that causes COVID. Our approach is going to take a broader test for all the genetic material that could be present from that virus that causes COVID, and we can detect, we anticipate any other genetic material from any other virus that could be in the respiratory tract.
So why are genomes so important when it comes to the coronavirus or other infections?
So the genome refers to all of the genetic material that makes up, in this case, viruses. You and I also have genomes. That's all the DNA that makes up our bodies. And with genomes, we can do both detection, so we can test to see whether viruses are present or not. But in addition, it gives us very important information where we can compare those sequences to see how the viruses are spreading. So at the moment, I just checked this morning, there are 75,000 SARS-CoV-2 genomes sequenced from around the world. So this is unprecedented. We've never had an outbreak like this where we've sequenced so many genomes. And by comparing them and looking at the sequence, we can see how these viruses, all that are causing COVID, have been moving around the planet. We can start to see now how they're moving between provinces, between communities, and that helps us understand the transmission and how to better stop the virus. And so we can do that now with SARS-CoV-2. With our technology that we'll be deploying, we can do that same approach with other viruses as well.
Very interesting. So how far do you think we are from rapid tests and rapid results?
So rapid tests, we have them. The technologies already being deployed by public health labs across Canada are very fast and very sensitive. So you will find out within 24 hours if there is a SARS-CoV-2 virus that could cause COVID in an infected individual. Our approach is going to be a little slower at the start compared to that. We can't do a 24-hour turnaround, but to give you an example, currently, if you have a cough or a sneeze, as you said at the intro, it might not be the SARS-CoV-2 virus. And currently, if you were not infected with that, then the next day, your sample would be tested for the flu. And if that was positive, the next day it would be tested to determine potentially what flu type it was. With our approach, we'll be able to tell all of those, answer all those questions all at the same time. So ultimately, that can get a lot faster.
Very interesting stuff, Andrew. Thanks so much for your time.
Thank you very much.